So we were just talking about Mendel's four postulates and how we could observe the segregation model that he came up with using a math trick known as a Punnett square. The Punnett square is mostly used for genetics now, but it can be used for any kind of where you have to figure out different combinations. So we're going to use a capital letter to represent dominant alleles and a lowercase letter to represent recessive alleles. So we'll just stay with the flower color. So we'll use P for purple, capital P for purple, and small p for the white flowers. So we have a true breeding purple and a true breeding white. And if they're true breeding, then they are what we call homozygous, which is both letters are the same. Capital P for true breeding purple or homozygous dominant. And small p for true breeding white or homozygous recessive. So when we cross, what we do is we figure out what all the possible alleles could be from the purple and what all the possible alleles could be from the white. So all the possible alleles from the purple are capital P and capital P. And so we would have a capital P for purple and a small p for white. And so every single possible combination is going to be a capital P from purple and a small p from white. And so you would get all of your offspring would be big P, little p, or what we call heterozygous. So now in the F2 generation, we can take the big P, little p, and put those at one side of the Punnett square, and the big P, little p on the other side of the Punnett square. And so then we just carry these letters down, capital P, small p, capital P, and small p. And what we end up with is a 3 purples to 1 white, because the presence of a capital P obscures the small p. And so all you have to do is just look for anybody with a capital, and that one's going to be purple. And if you don't have any capitals, that one's going to be white. And so we notice that we have 3 purples and 1 white, but I also want you to notice that only one of these purples is homozygous. Obviously, the white is homozygous also. So this would be a true breeder and would give only white when self-fertilizing. This one would be a true breeder and only give purple when self-fertilizing. But these two are heterozygous, just like the F1 generation was. And so these two, if they were to self-fertilize, then they would also give a 3 to 1 ratio. So you're going to get a 3 purple to 1 white phenotypic ratio, but a 1 to 2 to 1 genotypic ratio. So let's go over some of these terms that I just used. Homozygous means both alleles are the same. Whether it's dominant or recessive, if both alleles are the same, we call that homozygous. Heterozygous means that the 2 alleles are different. One will be dominant and the other recessive. Homozygous are true breeding, but heterozygous are not true breeding. Okay? So all of your possible Punnett squares are listed here. And so we have, like homozygous dominant crossed with homozygous dominant is going to give all homozygous dominant. Homozygous recessive crossed with homozygous recessive is going to give all homozygous recessive. If we cross a homozygous dominant with a homozygous recessive, we're going to get all heterozygotes. Homozygous dominant and homozygous recessive will give all heterozygotes. We could cross a heterozygote with a homozygous dominant, and it doesn't matter if the homozygous dominant is on top or on the side, both of these are going to result in the exact same 50% homozygous and 50% heterozygous, homozygous dominant and heterozygous. Likewise, we could cross a heterozygote with a homozygous recessive, and we get 50% heterozygotes and 50% homozygous recessive. Okay? And lastly, we could have a het mating, and if we have a het mating, then we get one homozygous, two heterozygous, and one homozygous. So a 1 to 2 to 1 genotypic ratio, or 3 to 1 phenotypic ratio. Okay? So the word phenotype is just what is the actual trait? What is the outcome? Is it tall or short? Is it yellow or green pea? Is it purple or white flowers? Whereas genotype is the actual alleles. So what is the genetic makeup? For example, a purple flower could be homozygous dominant. So the genotype is homozygous dominant. Or it could be heterozygous. So the genotype would be heterozygous. But in both cases, the phenotype would be purple. Two different genotypes, same phenotype. The presence of the capital P tells you that it has to be purple. Okay? So we have a genotypic ratio of 1 to 2 to 1 on the het mating, but we have a phenotypic ratio of 3 to 1. 3 purples and 1 white, even though 2 of those purples are heterozygous, and 1 true breeding homozygous purple. And in both cases, you can tell which one is the homozygous recessive because it has a different phenotype. So if you had a purple flower, you wouldn't know whether it was homozygous or heterozygous. You wouldn't be able to tell. So how can we tell? Well, we could do a test cross. A test cross is where we take the unknown dominant homozygous or heterozygous, we don't know. We take that one and we mate it with a homozygous recessive. We take a history individual and we mate it with a homozygous recessive. Now there are two possibilities, right? If it is homozygous, then none of the offspring would be white because they would all be hets. But if it is heterozygous, then 50% would be dominant and 50% would be recessive. So we'd have those hets that had the dominant phenotype, and those homozygous recessives would have the recessive phenotype. And those are your two possibilities. So we would be able to tell whether it was a homozygous or a heterozygous. We can't tell by looking at the color of the flower, but if we do a test cross, we'll either get 100% or a 50-50 ratio. Now, the 3 to 1 ratio is true of monohybrid crosses, or just one trait, but it's also true of a dihybrid cross, where we're crossing two different traits. So let's say we were crossing a dominant, yellow is the dominant over green, and round is dominant over wrinkled, okay? So if we made this cross of a homozygous and a homozygous dominant, and homozygous recessive, then all of our offspring would be hets at both traits. So in independent assortment, it says that the shape of the pea does not care about the color of the pea. The shape of the pea does not care about the color of the pea. So we still get a 3-to-1 ratio, 3 yellows for 1 green, even though it's 12 and 4, it's still a 3-to-1 ratio. And we get a 3-to-1 ratio on round versus wrinkled, again 12 to 4, so a 3-to-1 ratio on round and wrinkled. But we actually end up with a 9 round yellow, a 3 wrinkled yellow, a 3 round green, and 1 wrinkled green. So it's actually a 9 to 3 to 3 to 1 phenotypic ratio. The genotypic ratio, unfortunately, is very complex. 1 to 2 to 2 to 1 to 4 to 2, these 2 wrinkled yellows, 2 to 2 to 1 to 1. So there's 1 of this genotype, 1 of this genotype, 1 of this genotype, 1 of this genotype, 2 of this one, 2 of this one, 2 of this one, and 2 of this one. So there's not 1 to 1 to 2 to 2 to 4 to 2 to 2 to 1 to 1 ratio genotypically, but phenotypically it's still a 3 to 1 yellow to green, 3 to 1 round to wrinkled, and a 9 to 3 to 3 to 1 for the combination. 9 round yellow, 3 wrinkled yellow, 3 round green, and 1 wrinkled green, or 9 to 3 to 3 to 1. Okay, so that's called the law of independent assortment. Round doesn't care about wrinkled, wrinkled doesn't care about round, purple doesn't care about tall, purple doesn't care about round. You know, all of these traits sort independently. We now know that this is not true. We'll talk more about that in chapter 15, but it's actually only true if they're on different chromosomes or very far apart on the same chromosome. Because chromosomes are a bunch of genes that get inherited together. The last video that we have for chapter 14 will be on some of the other possibilities, like incomplete dominance, or multiple alleles, or multiple factors, multiple traits coming off of one gene. So we'll talk about that in the next video.